Point number 4 is genetic mechanism of development of neuroendocrine tumor. Neuroendocrine tumors present a wide spectrum of malignant disease from rather benign to very malignant variants. Benign tumors are basically which are take place at only one. They do not spread, but malignant tumor spreads. The majority of these tumors are sporadic, but there are several familial inherited syndromes to consider, such as multiple endocrine neoplasia type number 1 and type number 2, von Hippel-Lindau syndrome, and tuber sclerosis and neurofibromatosis syndromes. The MN1 gene is mutated not only in the M1 families, but recent studies shows that more than 40% of sporadic pancreatic NETs harbor MEN1 gene mutations. The same study reported that ATRX and DAXX genes are mutated in a significant number of tumors, as are the genes encoding components of the mammalian target of the rapamycin, which is mTOR signal transduction pathway. These findings have implication for the new therapies that have been proved for the treatment of PNETs, such as tyrosine kinase inhibitor, sunitinib, as well the mTOR inhibitor everolimus. A small intestinal NET shows a less varied mutational pattern in that the majority of the genetic alterations are found on the chromosome number 18. There seems to be no difference between the sporadic and the familiar type of small intestine NETs. A wide range of genetic alternation has been described for the different subtypes of NETs, but the mechanics underlying tumor development are essentially unknown, except for MEN2, in which an activating mutation of the RET proto-oncogene drives tumor progression and affords a direct genotype and phenotype correlation. Genome-wide screening of different types of NETs can now be performed for a reasonable price and is likely to generate the new insights into the tumor biology and carcinogenesis in various subtypes of the NET. Detection of somatic mutations in the gastroenteropancreatic neuroendocrine tumors using the targeted deep sequencing. Mutations affecting the mechanistic target of rapamycin signaling the pathway for the frequent in the human cancer and have been identified in up to 50% of the pancreatic neuroendocrine tumors. Grade A evidence supports the efficacy of the mTOR inhibition and everolimus in the pancreatic NETs. Although a significant proportion of the patients experience disease stabilization, only a minority will show objective tumor responses. It has been proposed that genomic mutation resulting in the activation of mTOR signaling could be used to predict sensitivity to everolimus. Patients and methods. Patients with NETs that underwent treatment with the everolimus at our institution were identified, and those with the available tumor tissues are selected for further analysis. Targeted next generation sequencing was used to resequence 22 genes that were selected on the basis of documented involvement in the mTOR signaling pathway or in the tumor genesis of the gastroenteropancreatic NETs. Radiological responses were documented using the response evaluation criteria and the solid tumors. Here are the results. Six patients were identified and one had a partial response and four had a stable disease. Sequencing of the tumor tissues resulted in the median sequence depth of 667.1 with one fold of coverage of 95.9% to 96.5% and 10 folds covering of 87.6% to 92.2%. A total of 494 genetic variants were discovered, four of which were identified as pathogenic. All pathogenic variants were validated using Sanger sequencing were found exclusively in the menin-1 and death domain associated with the protein which is DAXX genes. No mutation in the mTOR pathway related genes were observed. Conclusions. Targeted NGS is a feasible method with a high diagnostic yield for the genetic characterization of the pancreatic NETs. A potential associated between the mutation in NETs and response to everolimus should be investigated by the future studies. The keywords are here is MDOR, PNET, biomarkers, and next generation sequencing. Here is our next heading, which is NUTYA germline mutation is associated with the small intestine endocrine tumors. The genetic behind the predisposition to small intestinal neuroendocrine tumor is largely unknown, but there is a growing awareness of familial form of disease. We aim to identify the germline mutations which is involved in the carcinogenesis of the SINEDs. The strategy including next generation sequencing of exome or a whole genome of the blood DNA and in selected cases tumors DNA from 24 patients from 15 families with a history of SIN8Ds. We identified 7 candidates mutation in 6 genes that were further studied using 215 sporadic SINET patients. The result was compared with the frequency of the candidate's mutation in 3 control cohorts with a total of 35,688 subjects. A heterozygous variant causing an amino acid substitution, which is PGLY396ASP, in the MUTY DNA glycosylase gene was significantly enriched in a SRT and AT patients and resulted in the odd ratio of 5.09. We also found a statistical significant difference in the age at diagnosis between the familial and sporadic SINATs. MUTYH is involved in the protection of the DNA from the mutation which is caused by the oxidative stress. The inactivation of this chain leads to the specific increase of guanine and cytosine, which is greater than thymine ratio adenine transformations in the DNA sequence and has been shown to cause the various cancers in humans and experimental animals. Our results suggest that the P, which is GLY396ASP in the MUTYH and the potentially other mutants in the additional members of the same DNA excision repair pathway, such as the OGG1 gene, might be involved in driving the tumorigenesis leading to the familial and sporadic SI needs. Here are the keywords which are related to our topic.